Okay, Chapter 15, Regulation of Gene Expression. Prokaryotes and eukaryotes alter gene expression in response to their changing environments. So gene expression, basically what that is whether they're making a protein or not. So you can turn on and off your genes, or the expression of your genes and the making of your proteins due to responding to different environments. Multicellular eukaryotes also develop and maintain multiple cell types. So remember that, for example, the human body, we have all different types of cells: muscle cells, skin cells, brain cells, liver cells, stomach cells, etc. Each one of those cells has a different function. So remember that the whole genome is found in every single cell of your body. Now, you don't want to have all your genes turned on in all your cells all the time. That would be wasteful. It doesn't make any sense. You would be wasting energy and resources. So basically, in particular cells, you just want genes to be expressed that are going to produce proteins that are going to be helpful in those cells, or proteins that will be exported from those cells, such as your endocrine cells. Gene expression is often regulated at the transcription stage, but control at other stages is important too. So here's a fish that can see equally well in both air and water. Not sure what happened there. Natural selection has favored bacteria that produce only the gene products needed by the cell. A cell can regulate the production of enzymes by feedback inhibition or by gene regulation. Gene expression in bacteria is controlled by a mechanism described as the operon model. So here we have a metabolic pathway. This is feedback inhibition. So what's happening here is we have a precursor molecule that gets converted to a second molecule via enzyme 1. That product gets converted to another product by enzyme 2, and then that product gets converted to the final product, tryptophan, by enzyme 3. Now, as tryptophan starts to build up in the cell, it's not necessary to make more tryptophan. That would just waste energy and resources. So tryptophan actually acts as a repressor, turning off enzyme 1 in the pathway. That's feedback inhibition. As the product starts to build up, that product shuts down the pathway, shuts down the synthesis of itself. A group of functionally related genes can be coordinately controlled by a single on-off switch. The regulatory switch is a segment of DNA called an operator, usually positioned within the promoter. An operon is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. The operon can be switched off by a protein repressor. The repressor prevents gene transcription by binding to the operator and blocking RNA polymerase. Okay, so remember, RNA polymerase is the one that's going to bind to the gene and transcribe the gene, bring in the complementary mRNA nucleotide. So if you have a repressor that's present where the RNA polymerase is supposed to bind, it's going to block RNA polymerase and it's going to turn the gene off. The repressor is the product of a separate regulatory gene. The repressor can be in an active or inactive form depending on the presence of other molecules. A co-repressor is a molecule that cooperates with a repressor protein to switch an operon off. For example, E. coli can synthesize the amino acid tryptophan. By default, the trip operon is on and the genes for tryptophan synthesis are transcribed. When tryptophan is present, it binds to the trip repressor protein, which then turns the operon off. Again, as tryptophan starts to build up in the cell, the E. coli don't need any more of it. So tryptophan shuts down basically the synthesis of itself. The repressor is active only in the presence of its co-repressor tryptophan, thus the trip operon is turned off if tryptophan levels are high. Okay, so here's what that looks like. So we have tryptophan is absent, the repressor is inactive, see the repressor right here, it's inactivated. It doesn't have the correct shape to bind to the operator, so it's off. As a result, the operon is on because RNA polymerase is able to bind to the promoter region and transcribe the genes. Now, the proteins that get made, these are polypeptide subunits that make up enzymes for tryptophan synthesis. So these enzymes, as a result of being produced, are going to produce tryptophan, and tryptophan is going to start to build up in the cell. So now, as tryptophan starts to build up, tryptophan is present. The repressor becomes activated because tryptophan acts as a co-repressor. So it binds to the repressor and activates it. That's what a co-repressor does. It binds to the inactive repressor and activates it so that the active repressor can bind to the operator. So now that you have a repressor here that's bound to the operator, that's going to block RNA polymerase from binding here, and it's going to turn off the gene. A repressible operon is one that is usually on. That's the trip operon that we just looked at. Binding of a repressor to the operator shuts off transcription. An inducible operon is one that is usually off. A molecule called an inducer inactivates the repressor and turns on transcription. So this is going to be the lac operon. The lac operon is an inducible operon and contains genes that code for enzymes used in the hydrolysis and metabolism, so breakdown, of lactose. By itself, the lac repressor is active and switches the lac operon off. A molecule called an inducer must come in and inactivate the repressor in order to turn the lac operon on. For the lac operon, the inducer is allolactose, formed from lactose that enters the cell. Enzymes of the lactose pathway are called inducible enzymes. Analogously, the enzymes for tryptophan synthesis are said to be repressible enzymes. Okay, so here we have the lac operon. So lactose is absent, so the repressor is active. The repressor is bound to the operator, blocking RNA polymerase from binding to the promoter region and transcribing the gene. Okay, so what do these genes code for? They code for enzymes that break down lactose. So if lactose is absent, then these enzymes, we don't need these enzymes to break down lactose. Okay, but as lactose starts to build up, allolactose, which is structurally similar to lactose, acts as an inducer. Okay, so this is what lactose becomes. It becomes allolactose. The allolactose binds to the repressor and inactivates it. It changes the shape of it so that it can no longer bind to the operator. As a result, RNA polymerase can bind to the promoter and transcribe the genes that create the enzymes that break down lactose. Inducible enzymes usually function in catabolic pathways. Their synthesis is induced by a chemical signal. Repressible enzymes usually function in anabolic pathways. Their synthesis is repressed by high levels of the end product. So the last one that you just saw, the lac operon, that's inducible. And the repressible one, that's going to be the trip operon. Regulation of the trip and lac operons involves negative control of genes because operons are switched off by the active form of the repressor. You can also have positive gene regulation. E. coli will preferentially use glucose when it is present in the environment. When glucose is scarce, CAP, catabolite activator protein, acts as an activator of transcription. CAP is activated by binding with cyclic AMP. You should remember that cyclic AMP is a second messenger functions as a second messenger in signal transduction pathways. Activated CAP attaches to the promoter of the lac operon and increases the affinity of RNA polymerase, thus accelerating transcription. When glucose levels increase, CAP detaches from the lac operon, and transcription proceeds at a very low rate even if lactose is present. CAP helps regulate other operons that encode enzymes used in catabolic pathways. Okay, so here is what that looks like. So E. coli prefer to use glucose. Well, if glucose is scarce and lactose is present, then E. coli are going to use lactose. Okay, so you have cyclic AMP, which binds to the inactive CAP, thus activating it. Okay, now the activated CAP binds to the promoter region and actually increases the affinity for RNA polymerase to bind to the promoter. And it transcribes the genes that are needed to make the enzymes that break down lactose. Okay, so that's if glucose is not there, and lactose is present, so E. coli are going to use lactose as its energy source. So it wants to speed up the production of the enzymes that are going to break down lactose. Okay, so that's the whole purpose of this, is to speed up transcription. And the active cap helps that happen by increasing the affinity for RNA polymerase to bind to the promoter. Okay, so now let's say that lactose is present, but glucose is also present. Well, what's going to happen is you have your inactive cap. So in other words, the lac operon is still going to be on because you do have lactose present, but E. coli will preferentially use glucose, okay? So you don't need the E. coli don't need to make the enzymes to break down lactose at a high rate because they're going to use glucose first, okay? It's not shut off, it's just not transcribed at a very high rate. All organisms must regulate which genes are expressed at any given time. In multicellular organisms, regulation of gene expression is essential for cell specialization. Almost all the cells in an organism are genetically identical. Differences between cell types result from differential gene expression, the expression of different genes by cells with the same genome. Abnormalities in gene expression can lead to diseases, including cancer. Gene expression is regulated at many stages. So basically, these are all the stages at which gene expression can be regulated. So looking at DNA in the nucleus, you can have chromatin modification. DNA unpacking involves histone acetylation and DNA demethylation. Okay, so remember that you can have histone acetylation, which unpacks the DNA and allows it to be transcribed more than if the histones were not acetylated. Remember, DNA methylation makes the DNA more tightly compacted. So DNA demethylation, taking off those methyl groups from the DNA, is going to unpack the DNA, which is going to increase the rate of transcription. Okay? We have RNA processing, where your introns get cut out and your exons get spliced together. And remember that different exons can be used to produce different protein products. And then once in the cytoplasm, you can have degradation of the mRNA once it has served its purpose. You can also have degradation of protein once those proteins have served their purpose. You can have protein processing, such as cleavage and chemical modification, and then also transport to cellular destinations, or even destinations outside the cell. In all organisms, a common control point for gene expression is transcription. The greater complexity of eukaryotic cell structure and function provides opportunities for regulating gene expression at many additional stages. The structured organization of chromatin packs DNA into a compact form and also helps regulate gene expression in several ways. The location of a gene promoter relative to nucleosomes and scaffold or lamina attachment sites can influence gene transcription. Genes with highly condensed heterochromatin are usually not expressed, because remember that the enzymes needed to transcribe the gene just simply cannot get at the DNA if it's so tightly coiled together. Chemical modifications to histone proteins in DNA can influence chromatin structure and gene expression. In histone acetylation, acetyl groups are attached to positively charged lysines and histone tails. This loosens the chromatin structure, allowing transcription of genes to occur at a higher rate. The addition of methyl groups actually has the opposite effect. This can condense chromatin and lead to a reduced rate of transcription. So here's histone acetylation. Remember, histones are the proteins that the DNA wrap around. DNA methylation is the addition of methyl groups to certain bases in DNA, usually cytosine. Individual genes are usually more heavily methylated in cells where they are not expressed. Once methylated, genes usually remain so through successive cell divisions. After replication, enzymes methylate the correct daughter strand so that the methylation pattern is inherited. This is known as epigenetic inheritance. So though chromatin modifications do not alter DNA sequence, they may be passed to future generations of cells. The inheritance of traits transmitted by mechanisms not directly involved in the nucleotide sequence is called epigenetic inheritance. Epigenetic modifications can be reversed, unlike mutations in the DNA sequence. So again, DNA methylation is an example of epigenetic inheritance. Chromatin-modifying enzymes provide initial control of gene expression by making a region of DNA either more or less able to bind the transcription machinery. Associated with most eukaryotic genes are multiple control elements, segments of non-coding DNA that serve as binding sites for transcription factors that help regulate transcription. Control elements and the transcription factors they bind are critical for the precise regulation of gene expression in different cell types. Okay, so here we have a eukaryotic gene and its transcript. So notice that going from DNA to the RNA, or the mRNA rather, you have your introns that have been cut out, your exons have been spliced together, and the mRNA has been given a cap and a tail. To initiate transcription, eukaryotic RNA polymerase requires the assistance of proteins called transcription factors. So remember, transcription factors, those are the usually proteins that are going to help RNA polymerase bind to the promoter region of the gene. They may also act as repressors rather. General transcription factors are essential for the transcription of all protein-coding genes. In eukaryotes, high levels of transcription of particular genes depend on interaction between control elements and specific transcription factors. Enhancers and specific transcription factors. Proximal control elements are located close to the promoter. Distal control elements, groupings of which are called enhancers, may be far away from a gene or even located in an intron. An activator is a protein that binds to an enhancer and stimulates transcription of a gene. Activators have two domains, one that binds DNA and a second that activates transcription. Bound activators facilitate a sequence of protein-protein interactions that result in transcription of a given gene. Bound activators are brought into contact with a group of mediator proteins through DNA bending. The mediator proteins in turn interact with proteins at the promoter. These protein-protein interactions help to assemble and position the initiation complex on the promoter. Okay, so what does that look like? So here we have enhancers and activators. Okay, these are distal control elements. So we have a DNA bending protein that allows the DNA to bend, and your activators and your enhancers to interact with a group of mediator proteins, and your transcription factors also come together, and this whole transcription complex allows RNA polymerase to bind to the gene, thus transcribing the gene. So they all work together basically in order for RNA polymerase to be able to bind and transcribe the gene. That's basically what that means. Some transcription factors act as repressors, inhibiting expression of a particular gene by a variety of methods. Some activators and repressors act indirectly by influencing chromosome structure to promote or silence transcription. A particular combination of control elements can activate transcription only when the appropriate activator proteins are present. So again, remember, in different cell types, you need to express different genes. Okay, so here we have a liver cell and the available activators. Okay, so what's happening here is the albumin gene is being expressed, which is needed in the liver cell. Crystalline, the protein product crystalline, is not needed in the liver cell. So there are no activators available in the liver cell in order to activate that gene. Pretty fascinating. Here's a lens cell, however. Okay, so a lens cell is going to need crystalline, so the activators for crystalline are present in the lens cell, but the activators for albumin are not, so the albumin gene is not expressed. Unlike the genes of a prokaryotic operon, each of the co-expressed eukaryotic genes has a promoter and control elements. These genes can be scattered over different chromosomes, but each has the same combination of control elements. Copies of the activators recognize specific control elements and promote simultaneous transcription of the genes. Transcription alone does not account for gene expression. Regulatory mechanisms can operate at various stages after transcription. Such mechanisms allow a cell to fine-tune gene expression rapidly in response to environmental changes. We've talked about alternative RNA splicing. Different mRNA molecules are produced from the same primary transcript, depending on which RNA segments are treated as exons and which as introns. Okay, so here you can see that coding regions 1, 2, 3, and 5 are used to create the mRNA. In here it's regions 1, 2, 4, and 5. So this is important because you can have one gene that can lead to the production of more than one protein, depending on which segments are used after RNA splicing in your mRNA. The lifespan of mRNA molecules in the cytoplasm is important in determining the pattern of protein synthesis in a cell. Eukaryotic mRNA generally survives longer than prokaryotic mRNA. Nucleotide sequences that influence the lifespan of mRNA in eukaryotes reside in the untranslated region at the 3' end of the molecule. The initiation of translation of selected mRNAs can be blocked by regulatory proteins that bind to sequences or structures of the mRNA. Alternatively, translation of all mRNAs in a cell may be regulated simultaneously. For example, translation initiation factors are simultaneously activated in an egg following fertilization. After translation, various types of protein processing, including cleavage and chemical modification, are subject to control. The length of time each protein functions in a cell is regulated by means of selective degradation. To mark a particular protein for destruction, the cell commonly attaches molecules of ubiquitin to the protein, which triggers its destruction.